Hello, students. Welcome to KENE'S Educare, a group of KENE solutions. This is part 5 of lesson number 5, the fundamental unit of life, that is the cell, right? So, आपको पता है that हमारी human body जो है that is fully made up of cell, and we have millions and trillions of cells inside our body, and when a group of cell come together, they makes up the tissue. When a group of tissue come together, they makes up an organ. When a group of organ come together and start performing their specific function, they makes the organ system. And once we have organ system which is being operated inside us, we say that a multicellular organism is being formed. And if a multicellular organism is formed, it means human being is functioning properly. So cell जो है that is the basic structural and functional unit of life that we are having, and we can say one statement out of here is that there can be no, like, there is no life without cell. If there is no cell, there will be no life which is going to exist there. Okay. And what else we saw, that Robert Hooke, he was the first one जिन्होंने सेल को डिस्कवर किया था in the year 1665. So when we go back to this much early period, that is 1665, okay, वहाँ पे आप देखोगे तो माइक्रोस्कोप was not even discovered, and at that time coming up with this concept of cell is such a great task that Robert Hooke sir has done. Okay, because अभी समझो ये जो इन्वेंशन है उनका सेल का, इट इज़ लाइक फोन नहीं है एंड देन टू हैज़ कॉन्टैक्टेड समवन विदाउट द फोन. ठीक है, माइक्रोफोन नहीं था फिर भी उनको सेल के बारे में, माइक्रोफोन बोल रही हूँ, यू कैन से माइक्रोस्कोप नहीं था, देन टू ही इज़ द वन who came up with the concept of cell during such an early period, because microscope का invention बहुत ज़्यादा बाद में हुआ है, and cell is one such thing जो आप देख सकते हो, but it is clearly visible only under a microscope. ठीक है, आपको कहाँ दिखता है cell? माइक्रोस्कोप के अंदर. And the components that we are studying about the cell, that is we have discussed on the plasma membrane, cell wall, nucleus, cytoplasm. Okay, so ये सारे जो parts है which are present in the cell, those things can also be seen only with the help of an electron microscope because of the size. And as we have said that size and shape हर एक सेल का डिफरेंट होता है, जैसे कि वी हैव डिस्कस्ड अबाउट द अमीबा जो है वो अपना शेप जो है वो चेंज करती रहती है. ओके, एंड इफ आई टॉक अबाउट द नर्व सेल, सो नर्व सेल्स हैव अ कांस्टेंट शेप, दे डोंट चेंज देयर शेप. Okay, so we have different kinds of cells that exist in different parts of our body, like we have nerve cells, we have brain cells, we have fat cells, and we have many more different types of cells which exist in our body, and each cell is having different function to perform, right? जैसे जैसे यू कैन से हमारा जो फैट सेल है उसका फंक्शन अलग है, देन हमारा जो लिवर सेल है, वी हैव डिफरेंट डिफरेंट टाइप्स ऑफ सेल्स इन आवर बॉडी, राइट? So every cell in our body has different function to perform. So here in this lesson we are discussing on the fundamental unit of life, that is the cell, right? सो हमने यहाँ पे सेल के डिफरेंट कंपोनेंट्स पे भी डिस्कस किया, वी हैव डिस्कस्ड अ लॉट ऑन थिंग्स, राइट? सो सेल इज़, यू कैन से, अभी हम जो ऑर्गन्स के बारे में डिस्कस करते हैं, हम डिफरेंट डिफरेंट सिस्टम्स पे जब डिस्कस करते हैं तो वहाँ पे वी आर नॉट अवेयर ऑफ सच थिंग्स, है ना, कि हमारे बॉडी में ऐसा सेल है, बिकॉज दिस इज़ वन सच टॉपिक जो आप डिस्कस कर रहे हो. यू कैन से जब आपने स्टडी अपना स्टार्ट किया था व्हेन यू वर स्मॉल, your mama taught you about ABCD. So when you are studying about the different systems that occurs into our body, like the respiratory system, the you can say digestion, reproduction. This all systems are further things. The basic unit, the basic thing that we have, that is our cell, because वही तो एक important चीज़ है जो नीडेड है, राइट? उसके बिना तो ये सारे सिस्टम्स ही नहीं चलेंगे, बिकॉज हमारा ऑर्गन भी तो सेल से बना है, हमारा टिश्यू भी तो इज मेड अप ऑफ सेल, राइट? सो सेल बिकम्स द मोस्ट इसेंशियल फंडामेंटल यूनिट ऑफ लाइफ दैट वी आर हैविंग, एंड दैट इज वाई वन स्टेटमेंट इज बीन गिवन that there is no life without cell. अगर cell नहीं है तो no life can exist without cell, okay? And he is the famous scientist Robert Hooke. Just imagine having his photo is such a great task. उस टाइम पे कैमराज भी नहीं थे, and he was the one जिन्होंने खुद का एक माइक्रोस्कोप डिज़ाइन किया, and you can say इन्होंने फिर वो cells को observe किया, and he that time called those as boxes cells. क्या बोला था उन्होंने? boxes cells. And since he didn't had like any of the scientists, they didn't had any knowledge on this cell, he was just visualizing the structure of the cell with something like the honeycomb, because honeycomb का little compartments होता है ना. So he that time said that it looks like an honeycomb apartment. There are little apartments which are present, and we can even resemble that honeycomb with that cork, and cork is something that we get from the bark of the tree. Okay, so this is how is what he was giving his explanation, and he was trying to explain everyone that we have something called as cell, which is the fundamental unit of life, and this cell is derived from a Latin word which means a little room. Cell का मतलब क्या है? A little room, and inside the cell we have small structures called as organelles which perform different specific functions. Organelles के पास क्या है? ऑर्गेनल इज अ स्मॉल स्ट्रक्चर जो सेल के अंदर प्रेजेंट होता है, एंड ये ऑर्गेनल्स का भी इट हैज सर्टन स्पेसिफिक फंक्शंस टू परफॉर्म, राइट? सो ऑल ऑर्गेनिजम्स दैट वी आर हैविंग, सारे ऑर्गेनिजम्स के अंदर या तो एक सेल होगा, or else we can have more than one cells as well, and all the tissues and organisms, they are composed of cells itself, and here the statement comes as there is no life without cell. Okay, then we discussed on different activities, and we came on to studying about the structural organization of a cell, where we saw that हमारे cell के पास कुछ special components हैं, and we call this special components as organelles. तो अब ये organelles या हमारा जो cell है, how it is organized, तो जब हम cell को देखते हैं under a microscope, we come to know three different features in every cell which have been present. एक होता है plasma membrane, एक होता है न्यूक्लियस, एक होता है साइटोप्लाज्म. सो दिस आर थ्री इंपॉर्टेंट मोस्ट एसेंशियल फीचर्स विच आर प्रेजेंट इन सेल. ओके, तो जो भी एक्टिविटीज़ सेल को परफॉर्म करनी होती है इनसाइड द सेल, एंड वॉट एवर काइंड ऑफ इंटरैक्शंस द सेल इज हैविंग विद द एनवायरनमेंट, मतलब सेल और एनवायरनमेंट के बीच का जो इंटरैक्शन है वो पॉसिबल क्यों है? That is possible because of these three different features which exist in the cell, that is the plasma membrane, nucleus and the cytoplasm. So these three important features are very important here. Okay, after that we started discussing about the plasma membrane, where we saw that plasma membrane क्या होता है. plasma membrane जो है वो outer most covering होती है सेल की, जो सेल के कंटेंट्स को सेपरेट करता है, बिकॉज सेल हैज समथिंग दैट इज प्रेजेंट इनसाइड एंड दैट इज समथिंग विच इज आउटसाइड, राइट? तो जो एक्सटर्नल एनवायरनमेंट है सेल का और इंटरनल एनवायरनमेंट के कंटेंट्स को सेपरेट कौन करता है? दैट इज बीन सेपरेटेड बाय द प्लाज्मा मेम्ब्रेन. ओके, बट प्लाज्मा मेम्ब्रेन इज समथिंग विच परमिट्स द एंट्री as well as exit of some materials. अब कुछ materials they are going to enter inside the plasma membrane, and कुछ materials they are going to move out of the cell. So ये movements कैसे भी नहीं हो सकते. Plasma membrane is a barrier, you can say, because it is a selectively permeable membrane. ये सारे materials को cell के अंदर जाने नहीं देगा. So there are some kind of things, like there is a special kind of movement of substance, like a question comes into our mind कि तो कोई भी सब्सटेंस सेल के अंदर कैसे जा रहा है एंड कोई भी सब्सटेंस सेल के बाहर कैसे आ रहा है, राइट? सो वी हैव सम सब्सटेंसेस लाइक कार्बन डाइऑक्साइड और ऑक्सीजन विच इज मूविंग अक्रॉस द सेल मेम्ब्रेन. कार्बन डाइऑक्साइड एंड ऑक्सीजन, हाउ आर दे मूविंग इनसाइड द सेल मेम्ब्रेन? बाय अ प्रोसेस कॉल्ड एज डिफ्यूजन. कैसे? डिफ्यूजन के थ्रू. नाउ डिफ्यूजन, यू आर वेरी वेल अवेयर ऑफ डिफ्यूजन में क्या होता है. डिफ्यूजन इज अ स्पॉन्टेनियस मूवमेंट, ठीक है? कार्बन डाइऑक्साइड या ऑक्सीजन का जो स्पॉन्टेनियस मूवमेंट होगा सब्सटेंस का, दिस मूवमेंट इज ऑलवेज गोइंग टू टेक प्लेस फ्रॉम अ रीजन ऑफ हायर कॉन्सेंट्रेशन टू अ रीजन ऑफ लोअर कॉन्सेंट्रेशन. ऐसा कभी नहीं हो सकता कि सब्सटेंस का मूवमेंट लोअर कॉन्सेंट्रेशन से हायर कॉन्सेंट्रेशन हो. इट इज़ नॉट पॉसिबल. ओके, सो यहाँ पे मूवमेंट कैसा होने वाला है? इट इज़ ऑलवेज गोइंग टू बी फ्रॉम हायर कॉन्सेंट्रेशन टू लोअर कॉन्सेंट्रेशन. ओके, so something similar happens in the cell as well. जैसे we have some substances like carbon dioxide. Now we are all aware of कि carbon dioxide जो है that is the cellular waste that we are having, and if it is a cellular waste it needs to be excreted out by the cell, right? So this CO2 carbon dioxide is going to get accumulated in high concentration inside the cell. तो हमारे सेल के अंदर हाई कॉन्सेंट्रेशन में कार्बन डाइऑक्साइड जाके एक्यूमुलेट करेगा. सो नाउ हियर बिकॉज ऑफ द डिफ्यूजन प्रोसेस विच अकर्स, व्हाट हैपेंस, बिकॉज कार्बन डाइऑक्साइड का कॉन्सेंट्रेशन इनसाइड द सेल इज हायर, this carbon dioxide का concentration is going to move out of the cell, and this occurs by the process of diffusion. Okay, so this was all with respect to carbon dioxide. Now when it comes to oxygen, when oxygen enters the cell by the process of this diffusion, we see that the level of concentration of oxygen inside the cell decreases. So here diffusion, it plays a very important role in gaseous exchange, because carbon dioxide and oxygen, they are gases, right? So diffusion is going to play a very important role in gaseous exchange which takes place between the cell, as well as the gaseous exchange that takes place between the cell and its external environment. Okay, so water also obeys the law of diffusion. So not only we have carbon dioxide, so not only carbon dioxide and oxygen, they are involved with respect to diffusion and osmosis. Like osmosis, you'll come to know, because movement of a water molecule, it takes place through this semi-permeable membrane. Then we call this osmosis, right? Diffusion process ko, water is also one such molecule that is obeying the law of diffusion. Okay, so when you see that the movement of water molecule takes place through such a selectively permeable membrane. Now selectively permeable membrane is the plasma membrane, right? So when the movement of water molecule takes place through the plasma membrane, we call that as osmosis. And osmosis may be like osmosis process takes place through this selectively permeable membrane, but here also the water is going to pass from higher concentration to a region of lower concentration. And ये flow कब तक होगा? higher से lower concentration तक का, जब तक equilibrium is reached. Okay, getting my point? So this is what we have discussed on diffusion and osmosis. And osmosis में we have three different kinds of solutions which are present. एक होता है hypertonic solution, hypotonic solution, एक होता है hypertonic solution, एक होता है isotonic solution. तो hypotonic solution में what happens is, suppose if the medium that we are having, which is surrounding the cell, that is having high water concentration than the cell. मतलब the cell which is having the water concentration is less than the outside medium. So here what happens is, because the outside solution is more, because it is very dilute, the cell will gain the water. The cell के अंदर the water concentration is low, right? So from the outside solution, the water is going to enter inside the cell by the process of osmosis, and such solution, when the water is entering inside the cell, we call this solution as an hypotonic solution. Okay, and here the water molecules, they are free to pass across the cell membrane, because I told you that cell membrane is selectively permeable. But in this case, because of osmosis, because the cell doesn't have that much of water, what happens, ये molecule जो है वो pass करेगा across the membrane in both the directions, but more water will come into the cell. Why? Because the water concentration inside the cell was lesser as compared to the environment. Okay, so more water is going to go into the cell and then it is going to leave the cell. Okay, so the net overall result that we have is the water is going to enter the cell, and now the cell is going to swell up. Okay, why the cell is swelling up? Because of the water entering inside the cell, and here we have hypotonic solution. Okay, second solution that we have is isotonic solution. अब यहाँ पे क्या होता है, if the medium has exactly the same water concentration as the cell, अब मेरे पास जो मीडियम है उसमें इनसाइड द सेल जो वाटर कॉन्सेंट्रेशन है दैट इज सेम, एज वेल एज आउटसाइड सेल जो वाटर कॉन्सेंट्रेशन है दैट इज ऑल्सो द सेम. So here there will be no net movement which is going to take place across the cell membrane, and such a solution we call it as an isotonic solution. Okay, and third solution that we have is hypertonic solution. अब hypertonic solution में क्या होता है, see, if the medium it has lower concentration of water than the cell, ठीक है, सेल के पास ज़्यादा वाटर है एज कम्पेयर टू द आउटर, यू कैन से to the external medium. So what happens, cell जो है it is going to lose it water and it is going to give it into the outer solution. So यहाँ पे the cell size is going to shrink, and this solution that we are having where the cell is losing the water by osmosis, we call this solution as an hypertonic solution. Okay, so I hope you are clear with this thing. The reason behind explaining it again is because this is one of the important thing जो आपको 10th में भी होगा, 11th में भी होगा, 12th में भी होगा. सो आई वॉन्ट यू टू बी क्लियर विद दिस 3 types ऑफ सोल्यूशन, बिकॉज osmosis and diffusion is one such topic जो आपको it will come like as and when you are moving in greater uh, as and when you're moving ahead, you are going to different standards, 10th 11th 12th में आप जाओगे तो this will be the points which will not be explained to you all. तभी आपको ये explain नहीं किया जाएगा what is diffusion, what is you can say diffusion क्या है, then what is osmosis, what is hypotonic solution, because it has been imagined that since this was explained to you all in 9th standard, the same thing will not be repeated in your 10th standard. So you should know the exact meaning. If I'm talking about hypotonic solution, what does that mean? What happens to the cell in hypotonic solution? What happens to the cell in isotonic solution? And what happens to the cell in hypertonic solution? So you should be aware of it. Okay. Now, after that, we discussed on the cell wall, where we saw that along with the cell membrane, plants also have another rigid outer covering. Now, animals do not have this cell wall. Okay, so plants के पास होता है ये rigid outer covering, which we call it as a cell wall. अब ये cell wall कहाँ पे होता है? It lies outside the plasma membrane. Where does it lies? Outside the plasma membrane. And the plant cell wall किससे बनता है? The plant cell wall is mainly composed of cellulose. And cellulose का काम क्या है? Cellulose एक ऐसा complex substance है जो हमारे plants को structural strength provide करता है. क्या करता है cellulose? हमारे plants को structural strength provide करेगा. So here we have another term called as plasmolysis which takes place. Okay. So plasmolysis में क्या होता है? When a living cell loses water through osmosis, that is there is shrinkage or contraction of the content of the cell away from the cell wall, then we call it as plasmolysis. Now this is same thing that we have seen for hypertonic solution. Hypertonic solution में भी cell का concentration was higher than the medium, right? So what the cell has done at during that time, cell ने क्या किया? It has released the water into the medium. As it was following the as it took place through osmosis, because higher region से lower region में ही pass होता है, right? So hypertonic में भी we saw because it is losing water there was shrinkage which was observed, right? So this phenomenon we call it as plasmolysis. And lysis का मतलब होता है breaking. क्या lysis का मतलब क्या होता है? break करना. Okay. Now you have to look at the cell wall. How does it exactly looks like? It is a tough covering that the plant cell has, but the animal cells do not have. Okay. Then another one activity after that. After discussing on cell membrane and cell wall, we had the third component as the nucleus. Now clearly look at the structure of the nucleus, how it is. It has a nuclear envelope, and this nuclear envelope जो है, it has two membranes. First is outer nuclear membrane, and second one is inner nuclear membrane. So these are the two different types of nuclear membranes which we have. Then on the outside we have this nuclear pore. Nuclear pore के through क्या होता है, जो भी सेल से बाहर जाना है, that is going to go out of the cell through this nuclear pore. This is the rough endoplasmic reticulum. Now we have discussed on endoplasmic reticulum in the last class, and we have seen there why it is called as rough endoplasmic reticulum and why it is rough, because of the presence of ribosomes. ठीक है, and ribosomes जो है they are mainly involved with the synthesis of protein molecule, right? And this is where we have this chromatin, and chromatin का हमें पता है chromatin जो है it is a thread like structure जो condense होके it forms a chromosome, right? So in a cell, अभी अगर cell divide करता है ना, तभी ही हमें यू कैन से जब सेल डिवाइड करेगा तभी ये क्रोमैटिन मटेरियल कंडेंस होके क्रोमोसोम बनाएगा. बट इफ द सेल इज नॉट डिवाइडिंग तो वहाँ पे हमारा जो डीएनए है दैट विल बी प्रेजेंट एज अ पार्ट ऑफ क्रोमैटिन मटेरियल इटसेल्फ. कैसे प्रेजेंट होगा हमारा डीएनए? क्रोमैटिन मटेरियल जैसे. इट मीन्स क्रोमैटिन मटेरियल जो है दैट इज विजिबल एज entangled mass of thread like structure. Okay, so chromatin material is going to be visible as a mass of thread like structure. Okay, then whenever the cell is about to divide, जब भी आप देखोगे कि सेल डिवाइड कर रहा है तो वहाँ पे जो क्रोमैटिन मटेरियल है, एज आई टोल्ड यू कि क्रोमैटिन मटेरियल कब अगर सेल डिवाइड नहीं करता है तो वहाँ पे डीएनए प्रेजेंट हो गया एज अ क्रोमैटिन मटेरियल. बट वंस द सेल स्टार्ट्स टू डिवाइड, इट इज अबाउट टू डिवाइड, ये क्रोमैटिन मटेरियल इट इज गोइंग टू गेट ऑर्गेनाइज्ड इनटू क्रोमोसोम. किसमें ऑर्गेनाइज हो जाएगा ये? into the chromosome. Okay, then nuclei we have, they are the specialized part which we are having of certain chromosome, and this nuclei they are going to carry out multiple copies of the DNA. क्या कैरी करेंगे ये? multiple copies of DNA, and once we are having this multiple copies of DNA we are going to use this DNA to synthesize the ribosomal RNA. ठीक है, हम क्यों यूज करेंगे इनको? to synthesize the ribosomal RNA. Now, as I said, our nuclei we have two membranes. So we have outer nuclear membrane and we have inner nuclear membrane, right? So nucleus has a double layered covering, and we call this double layered covering as nuclear membrane. Okay. Now if you can see that we have nuclear membrane के ऊपर we have certain pores which are present, right? So the nuclear membrane it has pores which allow the transfer of material from inside the nucleus. ठीक है, इट शुड बी प्रेजेंट फ्रॉम इनसाइड द न्यूक्लियस टू इट्स आउटसाइड. तो न्यूक्लियस के अंदर से जो भी मटेरियल हैज़ टू मूव आउटसाइड, दैट मूव्स थ्रू दिस न्यूक्लियर पोर्स विच आर प्रेजेंट. ओके, दैट इज न्यूक्लियस से साइटोप्लाज्म के अंदर अगर कोई भी मटेरियल ट्रांसफर होना है, so that occurs with the help of this nuclear pore. Okay, now our nucleus it has chromosomes which are visible as rod shaped structure. हमारा जो न्यूक्लियस है उसके पास क्या होता है? उसके पास क्रोमोसोम्स है जो विजिबल है एज अ रॉड शेप स्ट्रक्चर ओनली व्हेन द सेल इज अबाउट टू डिवाइड. जब हमारा सेल डिवाइड करने वाला होता है तब उसके पास ये क्रोमोसोम्स प्रेजेंट होते हैं विच इज विजिबल एज अ रॉड शेप स्ट्रक्चर. ओके, नाउ सी, नाउ हमारे क्रोमोसोम्स के अंदर क्रोमैटिन मटेरियल आपको पता है जब हमारा सेल डिवाइड नहीं करेगा तो डीएनए विल बी प्रेजेंट एज अ क्रोमैटिन मटेरियल. जब हमारा सेल डिवाइड करना स्टार्ट करेगा तो वो क्रोमैटिन मटेरियल जो है इट इज़ गोइंग टू गेट फॉर्म टू अ क्रोमोसोम. अब ये क्रोमोसोम के पास क्या होता है? क्रोमोसोम हैज़ ऑल द इंफॉर्मेशन like whatever information is related to inheritance of characters that takes place from parents to offspring, वो हमारे पास प्रेजेंट होता है इन द फॉर्म ऑफ डीएनए, दैट इज डीऑक्सीराइबोज न्यूक्लिक एसिड के फॉर्म में प्रेजेंट होता है. एंड द क्रोमोसोम दैट वी हैव, they are composed of DNA and protein. This I have explained you all. हमारा क्रोमोसोम किससे बना है, डीएनए और प्रोटीन से बना है. एंड दिस डीएनए मॉलिक्यूल दैट वी हैव, उसके पास सारा इंफॉर्मेशन होता है विच इज़ नेसेसरी फॉर कंस्ट्रक्टिंग द सेल एज वेल एज ऑर्गेनाइजिंग द सेल. And the functional segment of the DNA that we have, we call that functional segment as genes. तो अगर कोई ऐसा सेल है जो डिवाइड नहीं करेगा, वहाँ पे डीएनए कैसा प्रेजेंट होता है, it is present as a chromatin material, and it is like this chromatin material वो organize कब होगा chromosomes में, when the cell is about to divide, that time. So the nucleus that we have, that plays a central role in cellular reproduction, that is to increase the number of cells. Nucleus plays a very important role जहाँ पे the process by which a single cell divides and it forms two new cell. Okay, so nucleus is playing a crucial role, crucial part along with the environment. It is determining the way the cell is going to develop, and it is going to, like, it will exhibit at maturity, as well as by directing some chemical activities of the cell. So nucleus is helping our cell in one or many other ways, right? So कुछ organisms जैसे कि बैक्टीरिया हो गए, ओके, बैक्टीरियाज में द न्यूक्लियर रीजन ऑफ द सेल वो पुअरली डिफाइंड होता है. व्हाई इज इट सो? बिकॉज it is not having nuclear membrane. Why the nuclear region is poorly defined? Because of the absence of the nuclear membrane. And when we are having such an undefined nuclear region which is only having nucleic acid means DNA and RNA. So अगर वहाँ पे nuclear region नहीं है, it is only having DNA and RNA, we call it as nucleoid. And such organisms जिनके पास nuclear membrane नहीं होता, उनको हम प्रोकैरियोट्स बोलते हैं. बिकॉज प्रोकैरियोट्स का मतलब क्या है? प्रो मीन्स प्रिमिटिव और प्राइमरी, एंड कैरियोट्स मतलब कैरियन विच मीन्स न्यूक्लियस. सो प्रिमिटिव न्यूक्लियस. So the organism who are not having a nuclear membrane, they are called as prokaryotes, and who are having a nuclear membrane, they are called as eukaryotes. Okay, so I hope this much is clear. So here you get some of the differentiating point between prokaryotes and eukaryotes as well, right? So prokaryotic cell, they also lack most of the other cytoplasmic organelles which are present in the eukaryotic cell. Now eukaryotic cell के पास और भी ऐसे cytoplasmic organelles present है जो prokaryotic cell के पास नहीं है, and there are many of the functions of such organelle which is being performed by the poorly organized parts of the cytoplasm. प्रोकैरियोटिक सेल के पास बिकॉज ये साइटोप्लाज्मिक ऑर्गेनल्स नहीं है, सो देर आर मैनी फंक्शंस विच द प्रोकैरियोटिक ऑर्गेनिजम्स कैन नॉट परफॉर्म बिकॉज उनका साइटोप्लाज्म ही वेल ऑर्गेनाइज नहीं है, राइट? एंड द क्लोरोफिल दैट वी हैव इन दिस फोटोसिंथेटिक प्रोकैरियोटिक बैक्टीरिया, दिस क्लोरोफिल इज एसोसिएटेड विथ मेम्ब्रेनियस वेसिकल. क्लोरोफिल किससे एसोसिएटेड है? membranous vesicle से associated है, and this membranous vesicle is going to be a bag-like structure, but not with the plastids as in eukaryotic cell. Okay, so this is the diagram of the prokaryotic cell which has been given, where you need to show the cell wall, the plasma membrane, ribosomes and nucleoid. Nucleoid किसको बोलते हैं हम? only with the nucleic acid and they don't have nuclear membrane, then we call that as a nucleoid. Says undefined nuclear region containing only nucleic acid. Now this nucleic acid can either be DNA or RNA, but it is not having a nuclear region, then we call that as nucleoid. So prokaryotic cell has a nucleoid in the center. Okay, after that हमने cytoplasm पे discuss किया, where we saw that when we look at the temporary amount of the onion peel as well as the human cheek cell, तो हम क्या देखते हैं? वहाँ पे एक large region होता है हर एक cell के अंदर which is being enclosed by our cell membrane. और ये region जो है, when you stain it with, अगर आपको onion peel वाला activity याद है, तो वहाँ पे we stain that with the saffranin stain, right? saffranin stain से हम उसको stain करते हैं with a drop of saffranin. So that is used for staining that onion peel. But there is a large portion of each cell which is enclosed by this cell membrane, and ये जो region है that takes up very little stain, वो बहुत ही कम stain लेता है, and we call that region as cytoplasm. क्या बोलते हैं हम उसको, साइटोप्लाज्म. नाउ साइटोप्लाज्म इज नॉट एनी ऑर्गेनल, इट इज़ अ फ्लूइड कंटेंट जो हमारे प्लाज्मा मेम्ब्रेन यानी कि सेल मेम्ब्रेन में प्रेजेंट होता है. साइटोप्लाज्म क्या है? साइटोप्लाज्म इज द फ्लूइड कंटेंट which is present inside the plasma membrane and it also contains many specialized cell organelle. तो हमारे साइटोप्लाज्म में और भी बहुत सारे स्पेशलाइज सेल ऑर्गेनल्स प्रेजेंट होते हैं which perform their own functions. So each of this organelle they are going to perform a specific function for the cell, and this cell organelles, they are all the cell organelles that we are having, they are enclosed by a membrane. ठीक है. So if I talk about prokaryotes, so in prokaryotes, beside the absence of a defined nuclear region, now we all know that prokaryotes do not have a well-defined nuclear region. They also don't have any membrane-bound cell organelles within them. So this is also one of the differentiating point कि prokaryotes के पास membrane-bound cell organelles नहीं होता, but eukaryotes के पास they also have a well-defined nuclear region as well as they also have a membrane-bound cell organelles which are present within them. Okay. So the significance of the membrane we can illustrate that with the help of viruses. किसके through? Viruses के through. Because viruses they lack any membrane and hence they do not show characteristics of life. Now viruses के पास एक भी membrane नहीं होता and that is the reason viruses they don't have any characteristics. They don't show any characteristics of life जब तक वो एक living body के अंदर ना जाए and they use its cell machinery to reply. Okay. Then we started discussing on this cell organelle. So I told you that whenever you are discussing on this cell organelle, आपको इसका diagram draw करना is important, right? तो ये जो आपको diagram दिख रहा है, you will have to draw that. So every cell has a membrane. अब कोई भी cell हो, every cell has a membrane around it so that it can keep its own content separate from the external environment. तो हर एक सेल के पास अपना खुद का मेम्ब्रेन होता है ताकि वो खुद के कंटेंट को अलग रख सके अपने एक्सटर्नल एनवायरनमेंट से. ओके. सो लार्ज एंड कॉम्प्लेक्स सेल्स इंक्लूडिंग सेल्स फ्रॉम दिस मल्टी सेलुलर ऑर्गेनिजम्स. तो हमारे जो लार्ज और कॉम्प्लेक्स सेल्स है यहाँ पे इट इंक्लूड्स ऑल द सेल्स दैट वी गेट फ्रॉम द मल्टी सेलुलर ऑर्गेनिजम्स एज वेल. हमारे सारे सेल्स को दे नीड अ लॉट ऑफ केमिकल एक्टिविटीज़ to support their complicated structure and function. तो हमारे सेल्स को क्या चाहिए? It needs a lot of chemical activities ताकि जो भी सेल का complicated structure और function है it can support that. Okay. So to keep these activities of different kinds separate from each other. अब ये activities को separate रखने के लिए, एक दूसरे से separate रखने के लिए the cell uses membrane bound little structure which is called as organelle. Cell क्या use करता है? Membrane bound structure which is called as organelles को use करेगा and this is one of the feature of the eukaryotic cell. ये क्या है? This is one of the feature of the eukaryotic cell जो distinguish करता है prokaryotic cell को eukaryotic cell से. And remember this thing that some of the organelles they are visible only with an electron microscope. And if we are not having any electron microscope, they are not visible to us. So some of the cells, they will be, you can say, visible only through this electron microscope. And we have already discussed about the nucleus, right? So nucleus is also one of the cell organelle. When you are writing an answer, even you can include nucleus here. Okay, so along with nucleus, we have some other important examples of cell organelles that we are going to discuss now. That is, we have endoplasmic reticulum, we have Golgi apparatus, we have lysosomes, mitochondria, plastids. Now, all the cell organelles are important because they carry out some very crucial functions in the cell. Okay. See, so we started discussing about this endoplasmic reticulum. Now endoplasmic reticulum is also represented as ER. So this endoplasmic reticulum is a large network of membrane bound tubes and sheets. Now if you see this structure, you can see here the smooth endoplasmic reticulum and rough endoplasmic reticulum. So जो smooth endoplasmic reticulum है, that appears like tubes, and rough endoplasmic reticulum, it appears like sheet. So that is why it has been said that endoplasmic reticulum जो है, that has like membrane bound tubes and sheets which are present, because it looks like a long tubules or round or oblong bags which are called as vesicles, and that is the reason we call this endoplasmic reticulum as a large network of membrane bound tubes and the sheets. Okay, so अगर आप endoplasmic reticulum का membrane देखोगे, you will see that this endoplasmic reticulum का membrane is similar to that of the plasma membrane. कैसा है? It is similar to that of plasma membrane, and we have two different types of endoplasmic reticulum. The first one is rough endoplasmic reticulum, that is RER, and second one that we have is smooth endoplasmic reticulum, that is SER. Okay, so now this rough endoplasmic reticulum, it looks rough under a microscope because it has a particle called as ribosomes which are attached to its surface. तो जब आप rough endoplasmic reticulum को microscope के अंदर देखोगे, you will feel like that it is appearing to be very much rough under a microscope. Why is it so? Because अगर आप देखोगे यहाँ पे, the rough endoplasmic reticulum has some dots on it, and those dots are the ribosomes. So because they are having these ribosomes which are attached on the surface, that is why this rough endoplasmic reticulum, it appears to be rough under a microscope. Okay, so the ribosomes which are present on rough endoplasmic reticulum, they are present in all active cell, and ribosomes are the sites of protein manufacture. हमारे body में protein manufacture कहाँ से हो रहा है? Ribosome से हो रहा है, and whatever manufactured protein that we are having, we are sending it to various places in the cell depending on which depending on which organelle needs that protein, and this is done using this endoplasmic reticulum itself. Okay, so now talking about the smooth endoplasmic reticulum. So here our smooth endoplasmic reticulum, it helps in manufacture of fat molecule or lipids, and this fat molecules, lipids, they are important for cell functions as well, because some of the proteins and lipids, okay, that is proteins we get from rough endoplasmic reticulum and lipids we get from smooth endoplasmic reticulum. So they help us in building the cell membrane that we are having, and this process where the proteins and lipids, they are helping us in building this cell membrane, we call this process as membrane biogenesis. क्या बोलेंगे हम? Membrane biogenesis. Okay, so some other proteins and lipids, they function as enzymes and hormones. तो हमारे कुछ और भी proteins and lipids जो present होते हैं cell के अंदर, they function as an enzyme as and hormones. Okay, so although the endoplasmic reticulum, it varies greatly in appearance in different cell, it always forms a network system. अभी अगर आप देखोगे endoplasmic reticulum को under a microscope, you will see कि वो बहुत ज़्यादा अलग दिखता है in the appearance as compared to different kinds of cell, because rough endoplasmic reticulum जो है, that forms a network system. So thus one function of endoplasmic reticulum is to serve as channel for the transport of material. तो endoplasmic reticulum का एक function क्या है? To serve as a channel so that it can help in transporting different materials and especially proteins between various region of the cytoplasm or between the cytoplasm and nucleus. तो अगर cytoplasm से nucleus तक हमें protein का transport करवाना है, so who is going to act as a channel there? एंडोप्लाज्मिक रेटिकुलम. या अगर हमें एक रीजन ऑफ साइटोप्लाज्म से दूसरे रीजन ऑफ साइटोप्लाज्म तक प्रोटीन का ट्रांसपोर्ट करना है, देयर ऑल्सो दिस एंडोप्लाज्मिक रेटिकुलम इज गोइंग टू हेल्प अस. ओके, so endoplasmic reticulum also functions as cytoplasmic framework. How does it functions? As a cytoplasmic framework where it provides a surface for some of the biochemical activities of the cell. ठीक है? So because it is functioning as a cytoplasmic framework, वो हमें एक surface provide कर रहा है for this biochemical activities which is being you can say engrossed by the cell. Okay, so if I talk about the liver cell, okay, So in the liver cells of group of animals which are called as vertebrates, vertebrates, okay, vertebrates के अंदर ये जो smooth endoplasmic reticulum होता है, that plays a crucial role. Now for what? Crucial role in detoxifying many poisons and drugs. तो जो अलग अलग पॉइजन्स और ड्रग्स को डिटॉक्सिफाई करने के लिए हु इज गोइंग टू हेल्प अस? Smooth endoplasmic reticulum is going to help us in detoxifying the poisons and drugs which are present. Okay, so this is the structure of the plant cell and the animal cells. And as I told you in the last class as well, that animal cells के पास you will see this centrioles are present, but plant cells में centriole नहीं होंगे. ठीक है, फिर आप देखोगे एनिमल सेल के पास सेल वॉल नहीं है, बट प्लांट सेल के पास सेल वॉल है, राइट. एनिमल सेल के पास क्लोरोप्लास्ट नहीं है, प्लांट सेल के पास क्लोरोप्लास्ट है. सो दिस इज व्हाट यू नीड टू डिफरेंशिएट, एंड यू शुड नो कि व्हेन यू आर ड्राइंग अ diagram or a structure on plant cell, what things are needed to be drawn and what things are not needed to be drawn. Okay, because the structure remains the same, it's only you have to draw. Because plant cell जो है that is octagon, I guess hexagon, 1 2 3 4 5 6 hexagon है, and animal cells are pentagon, you can say like that. You have to remember, you have to draw the structure. Okay, now the next component, the cell organelle that we have is Golgi apparatus. Next क्या है? Golgi apparatus. Now see, can you see the structure of Golgi apparatus? This is one of my favorite structure because आप इसको कैसे भी draw कर सकते हो. You just have to make it a wave like thing, right? बट साथ ही में आपको उसकी लेबलिंग भी पता होनी चाहिए. वेयर इज़ द इनकमिंग ट्रांसपोर्ट वेसल, वेयर इज़ द आउटगोइंग ट्रांसपोर्ट वेसल. ओके, ल्यूमेन कहाँ पे है, सिस्टर्नी कहाँ पे है, न्यूली फॉर्म्ड वेसिकल कहाँ पे है, एंड ट्रांसफैट कहाँ पे है. बिकॉज लेबलिंग इज़ वन इम्पॉर्टेंट थिंग विच यू शुड नो. तो आपने अगर ये 2-3 बार सारा डायग्राम प्रैक्टिस कर लिया ना फॉर द सेल, गोल्गी apparatus एंड ऑल, सो आपको ये बहुत ज़्यादा अच्छे से विल फिट इन टू माइंड. एंड डोंट वरी, वंस यू गो विद दिस 10th में भी इट इज़ गोइंग टू हेल्प you out some or the other way. So try to get this into your mind so that you never forget this in your future. Okay, so Golgi apparatus जो है, it was first described by Camillo Golgi. So it is named after the scientist who had described this. Golgi apparatus was first described by Camillo Golgi, and it consists of a system of membrane bound vesicle. Can you see this? It looks like this is a membrane bound vesicle which is present, which looks like flattened sacs. Okay, so this flattened sacs, they are arranged approximately parallel to each other in stacks which are called as cisternae. तो आपको ये जो अरेंज्ड यू कैन सी दैट ये जो सैक है, फ्लैटेंड सैक है, वो कैसे अरेंज्ड है? एक दूसरे से पैरेलली अरेंज्ड है, and they are arranged parallelly to each other in a stack, and we call that stack as cisternae. Can you see? So each stack or each sack that we are having, this is called as a cisternae. And the membrane, they often have connection with the membrane of the endoplasmic reticulum. Now the membrane that we are having, वो मेम्ब्रेन किससे, उसका कनेक्शन किसके साथ होता है? इट हैज़ द कनेक्शन विद द मेम्ब्रेन ऑफ एंडोप्लाज्मिक रेटिकुलम, एंड देयरफॉर it constitutes another portion of complex cellular membrane system. Now because it is having connections with the membrane of endoplasmic reticulum, that is the reason it is going to constitute another portion of the complex cellular membrane system as well. Okay, so the material which is synthesized near the endoplasmic reticulum, अभी endoplasmic reticulum के पास जो भी material synthesized है उसको पैक करेंगे, एंड इट इज़ देन डिस्पैच टू वेरियस टारगेट इनसाइड एंड आउटसाइड द सेल थ्रू द गोल्गी apparatus. तो गोल्गी apparatus के थ्रू क्या होगा? जो भी एंडोप्लाज्मिक रेटिकुलम के पास मटेरियल सिंथेसाइज हुआ है वो पैक किया जाएगा, एंड देन इट इज़ मूव टू द टारगेट, to various targets which are present inside and outside the cell, and Golgi apparatus is going to help in performing this function. Okay, so Golgi apparatus functions include storage. First of all, it is going to store this materials. Modification, so it is going to modify. Then packing of the product in the vesicle. Along with that, it is also going to pack the product inside the vesicle. And in some cases, complex sugars may be made from simple sugar in the Golgi apparatus. So suppose we have a simple sugar which is present in the Golgi apparatus. So Golgi apparatus can make that simple sugar or can convert that simple sugar into a complex sugar as well. Okay, so Golgi apparatus is also involved in the formation of, okay, so Golgi apparatus is also involved in the formation of what? It is involved in the formation of lysosomes. Okay, सो आई होप यू हैव अंडरस्टूड गोल्गी apparatus से क्या होता है. ये सिस्टर्ने के अंदर सारा मटेरियल आएगा एंड ये इनकमिंग ट्रांसपोर्ट वेसिकल्स आपको दिख रहा है, उसके थ्रू जो भी मटेरियल है विच इज़ सराउंडिंग द ईआर वो यहाँ पे आया इनकमिंग ट्रांसफर वेसिकल से. Then it is going to have a flow like this, movement like this through cisternae, and this is how we are going to get newly formed vesicle at the end of this Golgi apparatus, and this is the outgoing transport vesicles which have been present. And the main function of Golgi apparatus is storage, modification, packing of the product in the vesicle. This is the most important thing that the Golgi apparatus does. As well as Golgi apparatus simple sugar को complex sugar में भी convert कर सकता है, and it is also involved in the formation of lysosomes. Now from the screen I want you to write 4 functions of Golgi apparatus in your notebook. Come on, fast. Okay, I hope now you know the 4 functions of Golgi apparatus, and your homework for today is you need to draw all the diagrams that we are discussing on. Now see, he is Camillo Golgi, after whom the name Golgi apparatus has been kept, because he was the first one who have described about the Golgi apparatus, so it was named after him, Camillo Golgi. So Camillo Golgi was born at Cortino near Brescia in 1843. Where was he born? Near Brescia in 1843. And this Camillo Golgi, he has studied medicine at the University of Pavai. So he was studying about medicines and all at the University of Pavai, and after graduating in the year 1865 he graduated. He continued to work in Pavia at the hospital of Saint Matthew. So as he was studying on medicine, so graduation होने के बाद he was still continuing his studies, and he was you can say that he was doing an internship at the, he was working at Pavia at the hospital of Saint Matthew. And at that time most of his investigations were concerned with the nervous system. See, where nervous system is and where Golgi apparatus is, right? So इनका investigation किस पे था? Camillo Golgi, he was mostly concerned with investigating about the nervous system. So in the year 1872, कौन से year में? 1872 में he accepted the post of chief medical officer at the hospital for the chronically sick at Ebitio Grasso. So now 1872 में after working in that hospital, इन्होंने he accepted the position of chief medical officer at the hospital, and he first started his investigation into the nervous system in a little kitchen of this hospital, which he had converted into a लैब. तो उन्होंने क्या किया था, जो हॉस्पिटल का एक स्मॉल पार्ट था उसको उन्होंने लैब में कन्वर्ट किया था, बिकॉज ही वॉज इन्वेस्टिगेटिंग इनटू नर्वस सिस्टम थिंग. सो उन्होंने जो छोटा सा किचन था हॉस्पिटल का उसको लैब में कन्वर्ट कर दिया था. ओके, सिंस ही वॉज द चीफ मेडिकल ऑफिसर नो वन कैन से एनीथिंग टू हिम, CMO. ओके, so see, however The work of the greatest importance with Golgi, the most important work that Golgi had carried out, was a revolutionary method of staining individual nerve and cell structure, and this method is referred to as a black reaction. So उन्होंने जो छोटा सा स्मॉल किचन था जिसको उन्होंने लैब में कन्वर्ट किया था, he had made use of it, and he had given us a revolutionary work. So what kind of revolutionary work we have? So उन्होंने क्या किया, हम सबके नर्व को, what he has done, उन्होंने नर्व को स्टेन करने में हेल्प करवाया. लाइक इट वाज पॉसिबल बिकॉज ऑफ हिम दैट वी कैन स्टेन इंडिविजुअल नर्व एज वेल एज द सेल स्ट्रक्चर, एंड दिस मेथड इज व्हाट वी रिफर टू एज ब्लैक रिएक्शन. क्या बोलते हैं हम इसको? इस मेथड को हम बोलते हैं ब्लैक रिएक्शन, वेयर वी कैन इवन स्टेन द nerve of any individual. Okay, so this method, it uses a weak solution of silver nitrate and is particularly valuable. अब ये जो method है black reaction या staining an individual nerve, वो एक weak solution use करता है silver nitrate का and is particularly valuable. Now why this weak solution of silver nitrate is used? Because it is valuable. This silver nitrate is valuable in tracing the process, as well as it is one of the most delicate ramification of the cell. What it is? It is one of the most delicate ramifications of the cell that we are having. So all through his life he continued to work on this line. तो पूरी life जो थी he had spent it on, and he has continued to work on this ramification of cell and tracing processes and all. So he was modifying and improving this technique which he was developing. So for the rest of his life he was working on this, modifying and improving this technique, and Golgi received the highest honor and awards and recognition of his work. So because of the efforts and the hard work and the consistency that he has shown, Golgi, that is Camillo Golgi, he had received the highest honors and awards, and he was recognized for his work. So he shared the Nobel Prize in 1906 with Santiago Ramon Cajal for their work on the structure of the nervous system. So along with the Santiago Ramon Cajal, they were also the one who was working on nervous system. So he was sharing the Nobel Prize in 1906 along with them because of the efforts that Camillo Golgi has put on. Okay. So you don't know ki what work that you are doing now is going to take you to what kind of level in the future. We are not aware of it. What we need to do is, consistency is very much important part that we need to have, because if we are consistent with something, then there will be a result for sure, 100%. If not now, after 5 years, after 10 years, after 20 years, but the result will be seen. And same thing occurred with this Camillo Golgi. Whatever work he was doing, though it was like successful or not, it has not been given here, but consistency has led him to get this Nobel Prize, which was shared along with this fellow, like fellow students हम किसको बोलते हैं, that is also an honor. Like along with one student there are other students as well in a group. Then we can, आपने सुना होगा फेलो स्कॉलरशिप के बारे में भी. There are different kinds of scholarship. Like अभी पढ़ना is very much easy. Educating ourselves is not difficult right now. But when we go back to 1840s or 1800s में, उस time the education system I guess was very much strong, but people were not able to collect those knowledge some because of some or the other reason. कभी भी war हो रहा है, कभी भी कुछ हो रहा है. So there were many issues that the people had to face, and then they were able to study so hard. But the education that they had received, we cannot receive it. Okay. Now the next that we are going to discuss on is lysosomes. Okay, so this is the anatomy of the lysosome which has been given, where lysosome has a lipid bilayer, it has a plasma membrane which is also called as the cell membrane. Cell membrane के अंदर क्या है? Lipid bilayer है. Then we have this hydrolytic enzyme mixture. क्या है? Hydrolytic enzyme mixture. Outside to it we have this glycosylated membrane transport protein. Okay, so this is how the anatomy of the lysosome looks like. So this is the third cell organelle that we are going to discuss on. So structurally if you see, Lysosomes are membrane bound sac which are filled with digestive enzyme. Lysosomes क्या है? They are membrane bound sac जो किससे filled है? Digestive enzyme से filled है. Okay, so this digestive enzymes they are made by रफ एंडोप्लाज्मिक रेटिकुलम. ये डाइजेस्टिव एंजाइम्स किससे बनता है? रफ एंडोप्लाज्मिक रेटिकुलम से. एंड लाइसोसोम्स आर अ काइंड ऑफ वेस्ट डिस्पोजल सिस्टम ऑफ द सेल. लाइसोसोम्स क्या है? दे आर द वेस्ट डिस्पोजल सिस्टम ऑफ द सेल. सेल का जो भी वेस्ट होगा that is going to get eliminated through this lysosomes, and that is why we are calling this as the waste disposal system of the cell. Okay, so this waste disposal system or this lysosomes, they help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Now whatever kind of worn out cell organelles are present inside the cell, or whatever foreign material has been entered, वो सारा चीज़ें सेल से बाहर निकालने में who is going to help in clearing this all, or to keep this cell clean by digesting this foreign material and worn out cell organelles? Lysosomes are the one who are going to help the cell in this. Okay, so now foreign materials entering the cell जैसे कि such as bacteria or food as well as old organelles, they end up in the lysosomes, and lysosomes are going to break this complex substance into simpler substance. तो लाइसोसोम क्या करेंगे? जो भी कॉम्प्लेक्स सब्सटेंस होगा उसको ब्रेक करेंगे किसमें? सिंपल सब्सटेंस में ब्रेक करेंगे. ओके, एज वेल एज व्हाट आर दे गोइंग टू डू? अभी सपोज हमारे सेल के अंदर कोई भी फॉरेन सब्सटेंस या फॉरेन मटेरियल ऐड कर गया, जैसे हमने कुछ खाना खाया है, उसके थ्रू बैक्टीरिया हैज एंटर्ड आवर सेल. सो व्हाट द लाइसोसोम्स आर गोइंग टू डू? लाइसोसोम्स आर गोइंग टू digest that foreign material which has entered by cleaning, by keeping our lysosomes clean. So these are very much important, and whatever like old organelles we are having, that also end up into the lysosomes only, because lysosomes क्या करेंगे? They are going to break the complex substance into the simpler substance. Okay, and if lysosomes are able to do this, because अब lysosomes डाइजेस्ट कैसे करते हैं कोई भी फॉरेन मटेरियल को इनसाइड द सेल? बिकॉज दे हैव द powerful digestive enzyme within them which is capable of breaking down all the organic material. Okay, how the lysosomes are going to digest this foreign material? Because lysosomes के पास powerful digestive enzyme present होता है, and this powerful digestive enzyme is capable that it can break down all the organic materials. Okay, so during the disturbance in the cellular metabolism, अब सेलुलर मेटाबॉलिज्म के टाइम पे अगर कोई भी डिस्टर्बेंसेस आ रही है, फॉर एग्जांपल व्हेन द सेल गेट्स डैमेज्ड, अगर हमारा सेल डैमेज्ड है तो लाइसोसोम्स दे आर गोइंग टू बर्स्ट एंड द एंजाइम्स आर गोइंग टू डाइजेस्ट देयर ओन सेल. तो यहाँ पे अभी सपोज मेरा सेल पूरा का पूरा सेल डैमेज्ड है, लाइसोसोम क्या करेंगे? वो खुद को बर्स्ट कर देंगे, एंड बिकॉज ऑफ द बर्स्टिंग ऑफ लाइसोसोम द एंजाइम्स आर गोइंग टू डाइजेस्ट देयर ओन सेल, and therefore as the lysosomes are bursting out because the cell is ruptured or the cells are damaged, the lysosomes are also called as suicide bags of the cell because of rupturing themselves. They are called as suicide bag of a cell. So this is the most common question which has been asked. Dash are also known as the suicide bag of the cell. So those are lysosomes. So lysosomes are also called as the suicide bag of the cell. Okay, so I hope you are clear with this endoplasmic reticulum, you are clear with the Golgi apparatus, and you are clear with lysozymes. Now your homework for today is drawing the diagram of this cell 2 times, drawing the diagram of plant cell and animal cells 2 times, drawing the diagram of Golgi apparatus and lysozymes 2 times, lysosomes 2 times into your notebook. Okay, because practicing the diagram is more important, and I want you all to focus along with the theory part. You should have an habit of drawing the diagram as well. Okay, so in the next class we are going to discuss on mitochondria. After mitochondria we have plastid, vacuoles, and then the cell division process which includes mitosis and meiosis. So the next part can be the last part for this lesson as well, where after completing this cell division we are going to discuss, like in short summary, what all things we have learned in this lesson: fundamental unit of life, and after that we're going to discuss on 10 questions which have been given, like we will be making a comparison table between the plant cell and the animal cell, then a comparison between prokaryotic cell and eukaryotic cell. After that we are going to write कि what happens अगर हमारा plasma membrane rupture कर जाएगा या break down कर जाएगा, so what is going to happen, and what will happen to the life of cell अगर Golgi apparatus नहीं होगा. So now you have written the four functions of the Golgi apparatus, and from that four functions you have to figure it out कि अगर हमारे पास Golgi apparatus नहीं होगा, so what is going to happen to the life of the cell. Okay, then which organelle is known as the powerhouse of the cell and why, this you will come to know in the next part. Then where do the lipids and proteins they constitute the cell membrane and they get synthesized, how does amoeba obtain its food, so we have already discussed this, right? What is osmosis, we have already discussed this. Then we have this one activity which we will be discussing at that time itself. And the last question that we have here is, which type of cell division is required for growth and repair of the body, and which type of cell division is involved in the formation of gamete. So this also you will come to know in the next class. Now I hope whatever we have discussed till now in this lesson, fundamental unit of life, is clear to you all. So now the rest of the part for this lesson, fundamental unit of life, we will study that in the next part. Okay, thank you everyone.